for coming today on a Friday afternoon. As I said in my greeting, this is the largest group that we have had so far on our webinars. So I just wanted to welcome all of you, especially Dr. Gamm, who is working on CHM research as well as other sight restoration therapies. So we're very honored to have you here today. I want to make sure that I do say right away that all of our webinars, which you can find on the YouTube channel of the CRF, which you can find on our website, curechm.org, our sponsors are Allergan and Biogen, and they awarded us a grant to provide all these webinars. So we always want to just do a very heartfelt thank you to them. So this is a webinar, and I'm sure most of you are zoomed out by now. I see many names of people who come to our other ones. Hello. Because it's a webinar, we're not going to have everybody engaged. So it will be a chat feature only, which means that's my job as a host. As Dr. Gamm presents things, if you have questions, send them on in, and I will share them with Dr. Gamm. We'll have a period of question and answering at the end. And if it's fitting, I might, go in between and ask them in between. We are recording this webinar. So in the next day or two, or on Monday, Tuesday, we'll have it up on the website and you can share it or you can watch it again to get more information. I think that's it for housekeeping. So just want to again say thank you to everyone for coming. Dr. Gamm is one of our favorites, and I'm not kidding about that. I'll let him share the story about how we were able to find each other. It's a special story. And again, we just want to say thank you and welcome. I'm going to turn it over to you.
All right, thank you, Beth. I appreciate it. So, so I'm Dave Gamm, and I'm here at the University of Wisconsin in Madison. And I've been here since, going on 20 years now. I did my residency here. I have an MD and a PhD. I did my schooling at the University of Michigan in Ann Arbor. I'm originally from Michigan, and then came here to do my residency and fellowship in ophthalmology and stayed ever since. In large part because around that time, the University of Wisconsin, Jamie Thompson, had discovered human embryonic stem cells and ultimately iPS cells. And so I thought that was an opportunity for me to learn from the folks who really invented the technology, and that's turned out to be very fruitful. And I've developed a lot of relationships within the university at first from our very extensive stem cell community, and then now worldwide. And I'm very happy to apply that to choroideremia. It is worth saying a little bit giving a little bit of background on how that happened, because as a pediatric ophthalmologist, I work with choroideremia, but I work with a lot of different forms of retinal degeneration. And of course, I'd love to be able to have a part in slowing down or reversing all of them if you could. But why choroideremia in particular? And that has to do with a gentleman that you're all familiar with, and that's Randy Wheelock. So I can't even remember how many years ago it was, but it was at one of our ARVO conferences, the large Ophthalmology and Vision Research Organization meetings. And I was just walking through, I think I was giving a talk, and I was walking through the poster halls, and all of a sudden somebody grabbed the back of my shirt sleeve and nearly spun me around. And it was this gentleman who apparently had been in my talk previously, and that I was hustling to get to my next thing too. And he stopped me and he introduced himself as Randy Wheelock, as part of, his son had choroideremia and was involved in the Choroideremia Research Foundation, and proceeded to tell me just why it would be so important to work with choroideremia. And I quizzed him. I said, obviously, I said, choroideremia is a very important disease, and it's one that I'm very interested in. But I have lots of other things that I've got pressing right now. And so he said, well, I know you got to go somewhere, but after that, let's have coffee. And I said, fine. And then I started to walk away. He goes, I got to get a hold of you though. So I gave him my number, and he followed up immediately. I think he followed up twice in the next hour. I wasn't even out of the next meeting when I got a text from him. So he just, that persistence, but it's more than just persistence. It was an intelligent persistence. It was this he came from a position of really knowing the field and knowing, coming from essentially a non-scientific background, but still having delved into it enough where he made the argument. He would have made a very good lawyer too, because at the end I thought to myself, what have I just agreed to? But I've never looked back since, and it was one of the best decisions I made, not only because of the uniqueness of choroideremia in and of itself, but because of the people that I've met, starting with Randy, and then moving on to Eric and Chris and all the different folks that I've met through the Choroideremia Research Foundation, Corey. We had the Iron Man that came through, and I got to meet a lot of folks here at Madison. And it's just a really special group of folks and families. It's when you're dealing with degenerative diseases of the retina and inherited diseases of the retina, you're not really dealing with individuals, you're dealing with families. And that makes it very heartrending at times, but also very rewarding. Subsequent to that, then the CRF has been very generous in supporting the work that we do on two fronts, and I'm going to go into that a little bit during the course of my slide presentation, which I'll start now. All right, so I'm going to go ahead and share my screen, Beth. And you guys will all be very pleased to know that this is not a long presentation. This is not a lecture, but it's enough hopefully to give you a flavor of what we're doing. There we go. And hopefully that'll be a springboard for further discussion, because really what we want to do is use the time that we have to answer questions. And I have a number of questions that were given to me ahead of time that we'll address as time permits, but also allow you as participants to type in the chat things that come to your mind that hopefully I can provide at least decent answers for. Okay, Beth, can you just confirm that you can see this?
I sure can, and I'm glad that you asked me that. We have, it looks like 4 or 5 people who are calling in, so you can maybe describe the
Sure.
slides as well when you're talking about it. That would be great.
Yeah, absolutely. And in general, let me just get out of this here. In general, that's one of the things that you learn over time, which is that when talking to folks who have low vision and blindness, that it's kind of rude not to verbally describe things. So I will do that, but I do forget sometimes, and I'll say things like, as you can see here, or as indicated on your left. I sometimes forget that, and so if that does happen, please call me on it, and I'll continue to learn. Okay, so let's move. Okay, so what I'm showing here is just something to do. A lot of it's very, very basic, so I don't think necessarily anybody who isn't on looking at a computer screen or is necessarily missing anything. So there's not going to be anything so much revealing, but hopefully it will help those who do have access to a computer to follow along. So I'm showing just a picture of a head with an eyeball connected to the brain, and really what I use this for is to indicate that we're dealing with when we talk about the visual system, we're talking about everything from the front part of the eye all the way to the back of the brain, where the initial processing of vision takes place. And about 50% of your brain is used in developing visual percepts, or what you see as the outside world and recognize. And that can be important because in some cases, like in Leber congenital amaurosis, where people are born and really don't have a normal visual experience, even when they're very, very little, the brain portion of it doesn't develop normally. And so there are some concerns that therapies that take place even relatively early in life may not be all that effective because the brain isn't connected or plugged in too well with the eyes. So Luxturna and the RPE65 LCA trials, that was a concern there, which is, can you even get in early enough to affect the disease? Choroideremia, as the people on this call know, folks have good vision when they start out, or reasonable vision when they start out, which means that those connections are made to the brain. So that's a positive, that we're really looking at the problems that are occurring within the retina itself, and we don't have as much to worry about with what's going on behind the eyeball. So let's just concentrate on the eyeball now. And I'm showing a figure of a cross-section of an eye, showing the cornea and the lens on the left, all of the focusing machinery. And then you have this large cavity in the center of the eye called the vitreous cavity, which is filled up with jelly, sometimes has floaters that float around in it. And then in the very back of the eye, lining it like a wallpaper, is the retina. And the retina,
Dr. Gamm, I'm so sorry to interrupt you. What we're seeing is a very colorful, excuse me, protein.
You're not seeing? Oh, you're not seeing the eyeball, huh?
Nope, we're
Hold
not.
on,
We didn't see the head.
Let me stop sharing. And okay, you're looking at I got you on my wrong screen then, so I apologize for that.
That's okay. It was a really colorful one. It was
well,
nice.
My other one was even better, so I gotta restart that. So let me go with this one, and this should work this time.
Perfect.
Hold on a
Let's see.
sec here. So now do you see an eyeball?
Yep. There we go.
All right. Just because I put the effort into it, I'm gonna go back to the other one. So there's my head with the eyeball connected to the brain. Okay. Now you're seeing now there's an eyeball that I'm showing, and so that retina that lines the back of the eye is like a wallpaper. It's like a transparent wallpaper. And then behind the retina is the choroid, or the vascular tunic, or the waterfall of blood vessels that feeds particularly the RPE and the photoreceptors in the very, very back of the retina. So looking then at the retina itself in a stylistic form, like a cartoon type form, if you just peel off a retina, or if you see a retina that's detached, unfortunately, in the back of an eye during a surgery, it doesn't look like much. It just looks like a semi-opaque, thick piece of cellophane. But in actuality is comprised of multiple layers, like a layer cake, of different specific cell types, all of which have very elegant jobs to perform. So deepest within the retina is the retinal pigment epithelium, so a pigmented layer of cells that have a support function. And their function really is to remove garbage from the photoreceptors that accumulate, and to provide nutrients and oxygen and things like that, so that the photoreceptors themselves can operate at maximum efficiency. This is important because the photoreceptors in your body are, pound for pound, the most active cell type in your entire body. They're constantly firing, they're constantly detecting photons of light and converting those to signals that can be passed along to the next cell and the next cell. Because it's really a circuit. The retina is part of your brain. It's a very highly specialized part of your brain, but the retina is embryologically, during development, brain. It's made up of neurons, specialized neurons, but it's made up of neurons just like your brain is. And that's why when you think about replacing these cells or saving them or preventing disease, you can't really think of it like you might think of a degenerative disease of your joints or of your heart, which are muscles or bones or ligaments or cartilage. Really, the category that degenerative diseases of the retina fall into are more like Parkinson's and Alzheimer and ALS. So central nervous system or nervous system-based degenerative diseases. Unlike those other diseases, though, that affect the brain or the spinal cord, which are very hard and sometimes quite dangerous to go get at, and have effects that are very far away, the cell types that we're talking about in choroideremia are very, very localized and accessible with fairly straightforward, currently available surgical techniques. So a lot of the problems that you have in trying to create therapeutics for other diseases in the brain or spinal cord, we don't have in the eye. We also have better means of determining what happens after we do various interventions or therapeutics. So for those reasons, companies who are interested in developing therapeutics maybe for the brain will start in the eye, because it's more accessible, more likely to show an effect, and cheaper to do, and has less of a chance of causing serious side effects than going into the spinal cord or the brain. That's a good thing for us, because what it means is a lot of companies, whether or not they're interested in as their end game of being an eye-based company, still have to go through the eye to get to the brain. And that's great. The more people that have to deal with us, the better, because it means that that the folks here on this call, as well as myself, will benefit from that. So that's something to also feel good about, is that even when you hear about breakthroughs in therapeutics, even outside of the retina, a lot of times the retina is the canary that they use to get to where they're going. So in some respects, all research is good and all research helps. Of course, the more specific it is for choroideremia, the better. But it doesn't mean that when something comes through that doesn't have the disease choroideremia affixed to it, that it doesn't have value and applicability. Okay, so the two cell types that, it's called choroideremia, and this is clearly one of the conversations a lot of people have, because the choroid is the vascular tunic that feeds, that brings in all of the nutrients that the RPE then passes on to the photoreceptors, the rods and the cones that do the job of detecting light. But even though it's called that historically a lot because when the first people that looked into the eye, they saw a very, very pale retina. And they said, well, if it's pale, that means it doesn't have a lot of blood, which means it must have something to do with the choroid primarily. Nowadays, we predominantly think that the seat of the disease is in the RPE or the photoreceptors. And what happens is the choroid itself, when it's no longer has a job to do, so when the cells that it has to feed go away, it tends to shrink a bit. And it can undergo degeneration as well. But from other conditions like macular degeneration, it appears as though it can plump back up again if given the proper target cells to feed once, if in fact we're able to come up with a source, a replacement source of those cell types. So in general then, a kind of a sense of what genetic eye diseases do, and you've heard, there are hundreds of them just in the eye, much less, the thousands and thousands that are in the body. But in general, every cell in our body has a code and a genetic code. So we have 30,000 plus genes that run our different cell types. And depending on what the cell type is, it leans more heavily on certain types of genes than others. So for instance, there isn't really any other cell in the body that does exactly what the RPE cells or the photoreceptor cells do. So even though all the genes for making a photoreceptor or an RPE cell are in every one of our cells, only some of them are actually activated or used to make the structures within the cell so that cell can do its specialized job. So even though we have all the same DNA, a lot of that DNA is shut down depending upon what cell type you have. So the DNA itself is just a code, it's just a blueprint. What's important is the protein that particular gene makes, because that protein then is a physical thing that then interacts throughout the cell and performs a function. So you have this DNA to protein to function within each and every cell. So if you have a mutation in one of these genes, like in this case in CHM for choroideremia, then it can knock out or otherwise alter the function of that protein. Now again, if that piece of DNA is shut off, say in your liver cell, and it's not even used, then it doesn't have a problem in that particular tissue. But in the case of the eye, REP1, the protein that's encoded by CHM, has an important function of which we're not quite sure what it is. And I'll get back to that a little bit later. But if you have a mutation, then the protein isn't made, or it's made poorly, and the function goes away. Now, interestingly, if cells are dysfunctional, oftentimes what happens is over time, the whole cell will degenerate away. So this makes it important then to understand the time course of any degenerative disease. Because early on, when everything's functioning relatively normally and the whole circuit is still in place, then of course you want to be able to keep that as long as you possibly can. And then at some point, you might start to lose vision because critical parts of cells are no longer present or working appropriately, but the cell is still there. So it's not as though the cell is totally degenerated, it's just lost a critical component of its activity. At that stage, you still may be able to revive your own natural cells if there's enough of it left. And if enough of it's left, this is sort of like doing a defibrillator, right? If you defibrillate a piece of dirt, it's not going to come alive. But if you defibrillate somebody right after they had a heart attack, you might be able to get them back alive again. So, so that's kind of the sense, and that's kind of where gene therapy fits in. You still have to have a viable house or body to be able to revive in order for that to work. A little bit later on in the disease, if the cells have degenerated entirely, then you have two options. One would be to bypass it altogether by using prosthetic chips or maybe optogenetics, things like that, or replacing the cells that have been lost. And then even later on, where you have very, very late stage disease, you have a domino effect where now not only the cell types that were primarily affected in the disease, not only are they gone, but the remaining cells and tissue has become kind of disorganized or scarred. And so now you don't just need to replace one or two cell types, but you need to replace the whole tissue or maybe the whole organ. So where a particular person with a degenerative disease is on this continuum is very important. And so that leads to, as I alluded to just now, different therapeutic options. Early on, you want prevention. You want drugs if you can do it. If you can keep what you've got, it's better than replacing what you've lost. Later on, if you start to lose vision, but the cells are still present, you can think of gene therapy or possibly drugs. And then later on, if the cells are gone, the cell replacement or prosthetics. And then after that, whole tissue replacement, whole organ replacement, or maybe prosthetics that directly stimulate the brain, which are being worked on, although they have a lot of complexity to them. So what does our lab do? Well, our lab has two kind of basic arms that are directed towards choroideremia. One is to look at these somewhat earlier stages in disease so that people who are earlier in their course of disease would benefit from. And we have a Wheelock Award. Kim Edwards in my lab was the first awardee of the Randy Wheelock Award for research in choroideremia. And this is really trying to understand at a basic level what goes on. So we know the gene, we know the protein, but why is it that disruption of this particular protein, which is found in every cell type in your body, why does it specifically attack your outer retina? And we don't really understand that right now. And that limits our ability to come up with new therapies. Normally you want to uncover that black box because it could give us more shots on goal to develop better gene therapies and drugs and preventative approaches. So a portion of my lab then works on trying to better understand just normally what REP1 does, what this gene and protein does in photoreceptors and RPE using induced pluripotent stem cells. Now that's a more recent one. What we've been working on for a longer period of time is the concept of cell replacement or transplantation of cells that we can now create in very, very high volumes with very high purity and high authenticity in large 3-liter vats of cells. So we can make hundreds of billions of retinal pigment epithelial cells and photoreceptor cells, and we can tune them to cones or rods based on what we need or we want to do. And so we have a source of these human cell types now. So how do we go about making sure that they're safe and then delivering them and hopefully getting them to hook up appropriately? So I'll talk about that too. And that started in my lab and subsequently grew into a company called Therapeutics, of which I'm the chief scientific officer. And that's important because a lot of the things that we develop in the lab require a great deal of support in the order of sometimes tens of millions of dollars in order to get them to a clinical trial. And so organizations like CRF are important for providing the seed funds that allow us to start the experiments necessary for us then eventually to go to the deep pockets and to say, hey, we really think we've got something here that might work. And then they're the ones that evaluate that and turn around, and if it fits their business model and they think it's going to work, then they turn around and put a large investment into it. And we're seeing that right now, and so that's very encouraging. Okay, I'm going to concentrate on cell therapy. I was told that that's probably what a lot of folks are going to be interested in, although I'm very pleased to talk about our other project as well, looking at the function of REP1 in RPE and photoreceptors. So the first step in all this is, can we manufacture human retina? Because if the cells are already gone, if your RPE and photoreceptors are no more, then there's been a long, long history of folks making what I call wooden ducks, things that kind of, sort of, maybe look like the real thing, but only maybe 5%. But they're able to generate enough enthusiasm, and there's so much desperation that they can convince people that, oh, let's just wing it. Now let's just see if it works. This is a lot of the mesenchymal stem cell stuff, and people use the word stem cell just so loosely, and they use it a lot of times to try and confuse people into paying for things that they have no need to pay for, or trying to raise a buck in sometimes unscrupulous ways. So you have to be a kind of a buyer beware in this situation. But the fact of the matter is that we can manufacture human retina in the laboratory and now in clinical based companies like what we have now that are fit for human use. So what I'm showing on the screen now is what looks like these, almost like red blood cells, but they're yellowish in color. We call them golden Cheerios. And they're about the size of a large pinhead. And what we discovered many years ago was, and then patented, was a way in which we could take induced pluripotent stem cells, which start off as blood or skin cells, and then genetically reprogram them using a technology that was developed here at UW-Madison, make them into iPS cells, which have the capacity to make every cell type in the body, which is great, but also very difficult to control, because if you want to make a photoreceptor cell, you don't want to have one photoreceptor cell in a sea of bone or a sea of liver cells. What are you going to do with that? So you have to have a way to direct it towards the cell types that you're interested in, purify it out, and then make sure it's safe. And so we, a while ago, were able to devise a method in which we could separate out these early retinal structures from maturing stem cells derived from adult humans. And what we can do over time, and it takes a while, but these small little retinal organoids, we call them, or retinal tissues that we grow in the laboratory, make photoreceptors. They make RPE, and these photoreceptors respond to light, and in many respects look like near newborn rods and cones. And the kind of cool thing that's spinning around right now shows the little red dots are cones, and the green lines are the outer segments of rods. And if you look at the surface of one of these organoids, these retinal structures, you'll see these red dots, these red little protrusions that are the outer segments of cones in this particular image, and then the red dots deeper are the cells, the nuclei of the rod photoreceptors. So this was very, very exciting, and really was the impetus for us developing patents and then ultimately starting Opsis Therapeutics. I should also say that I have to declare that I do have an interest in Opsis Therapeutics. I was a co-founder and I'm chief scientific officer. So feel free to not believe anything that I say, but it's also not publicly traded, or there's no way for anyone to invest, and we're not looking for money anyway. So don't worry about that. But it is important for me to bring up, because I can't reach you. I can't reach patients if I sit in my lab all day. I have to be able to interact with individuals and companies and resources that allow for clinical trials to be generated and to move forward. And here's an example of some retinal pigment epithelium that we made from patients with various diseases, including choroideremia. We have iPS cells from individuals that do have choroideremia that we're using to understand the disease. So you can make them, that's fantastic. So you can make the spare parts, but just like if you laid out all the spare parts of a Boeing 747 on a runway, you'd say, great, all the parts are there, but you're not going to fly with all the parts just sitting on the runway. So can we take those parts and perhaps, hopefully, be able to get them to hook up with what's remaining in a patient? And so for that, we've looked at a lot of different ways to do that, not all of which require scaffolds or special delivery devices, but they may. And so we've worked with a wonderful group of engineers here at UW-Madison, led by Dr. Gong and Dr. Ma. And we've developed these patches, or these biodegradable, very, very tiny micromolded patches. And our latest generation is what we call an ice cube tray. So these are really, really, really tiny, as they must be in order to be able to be inserted under the retina. But what it allows us to do is it allows us to take the cells that we make, the RPE and the photoreceptors, and place them into a patch so that they have some organization to them. and that we can just direct them to a particular place within the retina as opposed to have them float or kind of go wherever they want to go. And so if we blow up one of these biodegradable micromolded patches that have already been seeded with photoreceptors, which are shown here in red, the little green dots are outer segments that they're showing. It's just like an ice cube tray. So you've got the ice cube tray itself, which is this biodegradable material that does well under the retina. And then the ice are the cells that we want to replace in choroideremia. I should say as well that these same cell types are what we want to replace in other diseases like macular degeneration. So when you hear about cell therapies for macular degeneration, pay attention to them, because the cell types that they're replacing or that we're replacing too in macular degeneration are the same cell types that we're looking to replace in choroideremia. So a win for macular degeneration and stem cell technology is a win for choroideremia. AMD, oftentimes I call it the 800-pound gorilla, but if you're riding on the shoulder of that 800-pound gorilla, you're as good as the gorilla itself. And here's an example. We collaborate with a group at the National Eye Institute named Kapil Bharti, who's starting a clinical trial for macular degeneration to replace RPE only. But we're working with him to do a combined patch with RPE and photoreceptors. And we use a model that he developed that relies on the pig. It's a pig model because pigs have an eye anatomy that's very, very similar to humans. And so it allows us to test these larger devices that are too big to put into a mouse eye or a rat eye. And so we have a close collaboration with Dr. Bharti, who also has a very strong interest in choroideremia and is interested in making pig models of choroideremia, which would be very, very valuable. But here's, I'm showing a video. This is one of our surgeons, Juan Amaral, injecting one of our patches into the subretinal space of a pig eye that has a degeneration of both photoreceptors and the RPE. So this is moving along very, very well. Okay, so that's really the end of my kind of brief overview of what we're doing. At this point, I'll kick it back to you, Beth, just to see if there's any questions. But then as people are formulating their questions and typing them into chat, I can also address some of the questions that were provided to me prior to today.
Sure. I think that sounds like a good plan. Right now, we don't have anybody that's been typing. I think everyone so far has been watching your totally excellent presentation. This was fascinating.
Well, you say that's off the boys' bath, huh?
Hey, here comes a question. I'll start you off with a question. And as other people are using that chat feature and sending those questions in, how about you answer this question from Don, and then you can maybe answer some of the questions that we sent in to you earlier. So so Don is asking, is this a particular kind of pig model?
No, so right now, there are no animal models, large animal models of choroideremia. And in fact, the pig, this is an issue in all types of therapeutic development. There is a lack of large animals that mimic diseases that humans have. So in pig, there's a type of retinitis pigmentosa, a P23H pig. And that was developed, but it's got a lot of problems with it. It's very variable. And we were actually looking into it, and the group that we wanted to work with, it was like $50,000 for a pig. So that's just not doable. So a lot of times what happens, and there's no non-human primate model, there's no canine model. There are naturally occurring dog models of some types of retinitis pigmentosa. The Briard sheepdog was used to advance Luxturna, the RP65 gene therapy product. But those are few and far between. So you can genetically engineer some of these mutations into large animals, but it costs something to do, and then you have to raise them and breed them and maintain colonies. So a lot of what folks do is to do, say, some sort of an intervention to eliminate the cell types that are affected in the disease, which is not a disease model per se. It just mimics the end result of that disease. So for instance, with the pig, what he did that ultimately led to the FDA approval for his clinical trial for RPE replacement in macular degeneration is a laser-based model. So these are normal Yucatan pigs that they're using a laser, which is painless. But the laser allows a degeneration of the RPE and overlying photoreceptors. So it's instantaneous. That makes it cheaper. It does result in the loss of the cell types that we see that are lost in choroideremia, but you don't have the secondary choroidal effects to them either. So it's kind of a poor man's macular degeneration, a poor man's choroideremia, or a poor person's, I should say. What we're trying to do is we're trying to say, well, we can now genetically engineer actually the choroideremia loss of function into a pig model, and then use all the facilities that are already present in the National Eye Institute. There's a surgery suite there for pigs that would rival most human surgery suites. I've been there many, many times. We actually use that facility to advance our cell therapeutics. And so plugging into that and having model systems that you can use and publish with is very valuable, because a lot of companies don't necessarily care about disease X, Y, or Z. They care about their product, and they want their product to get into humans, some humans somewhere, and then use that as a springboard to get more investments, and then to expand their operation to hit more and different types of diseases. What the gateway disease is oftentimes doesn't matter, and it can be an ultra rare disease. RPE 65, or Leber congenital amaurosis, very rare, but it didn't stop Luxturna from developing that therapy, and ultimately Roche from buying it for them for $4 billion. Roche, however, probably has their sights set on something bigger like hemophilia. So if we have nice, neat packages that are based around choroideremia, whether they be stem cell models or mouse models or pig models, then investigators will gravitate towards them because the tools are there. And then companies will gravitate towards it because the evidence will be there in the preclinical animal model studies. So that's a much longer, one thing that you're guaranteed is I will give you a much longer answer than you anticipated. But oftentimes the questions you ask, which are wonderful, are nice springboards, I think, into these just very general questions that people have on their minds that I've discovered over the course of the last couple of decades.
And treating so many patients or seeing patients in the clinic. Hey, we have a question from Debbie, and she's asking, in your timeline of therapeutic options, can you describe the difference between disruption and loss?
Yeah, so disruption, which just means that the gene is no longer working and so the function of that gene is no longer present. Let me give another, I love metaphors, so let me give you another metaphor / analogy. So if you have a house, and the house and the electrical system goes out, the electrical box goes out. It's cold, it's dark. The house is still there, it's still standing, but now you can't live in it. And if you can't live in it, then there's nobody there to keep it up. And so over time, the house falls into decay. If you get in there early enough and you fix the electrical system, the house, everything else there, the plumbing and the walls and everything else is still there. So so it's just go in and fix the electrical system if you can. If the whole house has just decayed and it's no longer there, a new electrical system isn't going to help you. You need a new house. So the idea of disruption and gene therapy is to get in and fix the electrical system before the house gets cold and dark and everyone moves away and it decays to nothing. Cell therapy is you walk into the neighborhood and the houses are gone. And so, it doesn't matter if you've got a big roll of electrical wire in your back pocket, you got to build new houses. And we can build new houses. The question is, can we set them up just right? And will they play nice with their neighbors? So that's kind of the fundamental difference between cell therapy, which is late stage intervention of degenerative disease, and gene therapy or drugs, which is earlier stage intervention. And the gene therapy aspect of it is ahead. And you guys all know that because there are 3 choroideremia gene therapy trials already. Cell therapy, because it's a much more complex thing, replacing an electrical system versus replacing a whole house. That's more recent. And so that's, necessarily lagged behind. People were doing gene therapy before anyone knew what a stem cell was, a human stem cell was. Our trajectory is faster than the earlier trajectory of gene therapy. So I don't think you have to necessarily look exactly at of how long it took for gene therapy to hit patients in terms of how long it will take cell therapies to do that. I think we've seen things shorten in terms of their time frame, but I'm also cognizant of the fact that time is time, and one day is too long for people who are dealing with devastating diseases. So when you say, hey, this is going so fast, it'll be here in 3 years, I don't say that anymore, or 2 years or 5 years or whatever it is. I mean, my reference might be, well, the last Tom, Dick, or Harry, or Jane that worked on this took 30 years. So yay for us, 5 years. That's fine if you're talking to another scientist who's doing a 30-year project, but it's not fine to a family who's dealing with progressive vision loss. So time is relative.
Hey, Dr. Gamm, can you talk a little bit more about that? Because we have a handful of folks here who are asking, this is something that you and I talked about yesterday, about timelines, about, you educated me that there isn't a natural history study per se. So can you just talk a little bit more about that? And then let me tag that on. We have another person that said that they weren't able to get into a gene therapy trial after all the process and the tests and the natural history study because they let me just read this, that the implant requires steroid use, which they felt was not recommended. So the question is, do you think at what point would this process be appropriate for CHMers in the advancement of the disease?
Okay, well, part of that I'll be able to answer, and part I'll have to get some more clarification, particularly on the steroid implant, because I don't know if they're talking about cell therapies, because there was a steroid implant for an AMD trial, but there have been no stem cell based therapies that have been put forward for choroideremia per se. So I don't know exactly. So I'll have to get some more clarification on that. With regard to the differences and the idea of natural history studies, natural history studies are always good to know. The more you know about a disease, the better. However, going back to my timeline, so if you're developing a gene therapy, you've got to be pretty sure that you still have some houses around, right? Because if you're bringing that electrical wire and there isn't a house to be found, you're doing the patient disservice because you're putting that potentially at harm's way, and there's no way that it's going to help you. For cell therapies, because it's agnostic, it doesn't care about the electrical system, the plumbing system, it's just bringing in a whole new house. It doesn't really matter to us it does to some degree, but in broad terms, it doesn't matter how you lost your house. Maybe it was the electrical system, maybe it was the furnace, maybe it was the plumbing, whatever it was. Doesn't matter. It's all gone. It's all got to be replaced. So in that regard, cell therapy is more agnostic and more of a broad-based approach. And that's why if it advances in any disease, it's helpful to choroideremia because it looks at it and goes, okay, I care that it's choroideremia because I have to think about the choroid too. And I have to think about how is your inner retina looking? Did your brain develop normally? Did the visual portions of your brain develop normally? The answer to that is yes. So it's not like you can be completely flippant about, oh, we'll stick it into anybody. But at the same time, with a gene therapy, it's only for that gene. And it's only if you're early enough in the disease where the house is still there. So it's much more specific. And some things like CRISPR-Cas9 or gene editing, it's not just the gene, it's the actual mutation in the gene. And there's over 140 different mutations in the CHM gene that can cause choroideremia. So that means if you're going to do gene editing, you might need 140 different therapies just for choroideremia. Now there are ways maybe to short circuit that, but that's getting into too much detail. But that essentially is the difference between longitudinal studies and the importance of them in gene therapy versus cell therapies. Getting to the timeline question, very, very good one, and one that I try to avoid as much as I possibly can because there's so much of it that we don't control. I will say that our current timeline has us doing photoreceptor and RPE replacement in 3 years. That's if everything goes well. That's if the FDA likes our data. That's if we have a partner through Opsis Therapeutics that will pay the $20 million that it needs to be paid to do it, which means that the FDA has to approve it. It means that no other cell therapy that's going on that's totally independent of what we're doing doesn't have some really bad effect that causes the whole system to say, whoa, wait a minute, we shouldn't be doing cell therapies. That happened in gene therapy. So the very first gene therapies were done in patients that had, in children, and they had the side effect of causing leukemia, and a couple of the kids died. That totally stopped gene therapy for over a decade until Luxturna came back on board. And the only reason that came on board was because it was an eyeball, and people said, well, you know, it's safer because if something goes wrong, we can remove an eye. We can't remove a brain or a heart or anything, or everybody's blood. Again, these are things that we can't control, that we can't necessarily foresee, that can affect the timeline. So I've learned a long time ago that if I say a number, I'm going to get emails later if we don't hit that number. But I can tell you at this point, at least in the work that we're doing, I'm usually conservative, and I'll say, you know, if I think it's going to be 3, I'll say 5 or 6. We have yet to hit a mark or a milestone later than what I thought. But now we're in a realm, and that's more on the research end of things, but we're now in a realm where we're reliant upon large companies or large organizations to be able to foot the bill for clinical trials. And so I can't control their business model and what they want to do or what they have the appetite to do. But right now there's a great deal of enthusiasm, and we've spoken to many companies, and we're actually reviewing a number of different things that, in a lot of different directions that we can go. So as things stand right now, I would expect to be able to do that. We will be pursuing in patients early stage trials in photoreceptor and RPE replacement, hopefully within 3 years or around 3 years from now. That's our timeline.
And I got to say that since we have a good, close working relationship with you, the best place to find out about those timelines would be going to the foundation, going to the CRF, because we'd be able to post updates.
Oh, yeah. Oh,
right?
Oh, yeah.
Right.
Yes. And again, I should also mention too, when I say photoreceptor and RPE replacement, you know, choroideremia as one of, so this gets into like phase 1, phase 2, phase 3 studies. Those initial studies will be what are called phase 1. They will involve perhaps 16 patients. And so those are for safety, really safety alone. And those first 16 patients may very well be folks that don't have choroideremia. They may have some other form of RPE or photoreceptor loss. That's fine. You don't necessarily need to be first in line. You know, somebody has to go forward and make sure it's safe. And then if it's safe and it shows promise, meaning people are actually helped by it, then it will get opened up to a larger group of phase 2 and then ultimately a phase 3, and then it becomes commercialized and available to everybody. So it's kind of these, you know, it's a step-by-step process that of course does take time, and that's frustrating in many regards, but it's there to make sure that things are safe. Just like we talk about COVID vaccines, right? I think we were all in the same boat that we would want that to be tried before it just was given to 3 trillion people on the planet. And then we find out 5 months later that there's a really bad side effect to it. So you do have to have these kind of smaller groups of you know, folks that have little to lose and have great courage to be able to test these out. And then lastly, you know, we don't expect things to be grand slam home runs right out of the gate. Just like any technology, we didn't start out with supersonic jets. We started out with the Wright brothers and just trying to survive running off of a sand dune. And then ultimately the technology showed promise, and then it got refined over time and better and better and better to where we are now. Same thing with computers and everything else. So, you know, any sort of technology, the key is to make sure it's safe, that it makes sense. Hopefully it shows improvement, and then that fosters additional investment and additional enthusiasm to go in and make it better and better and better and better. So again, I just want to make sure that we have that I'm coming off with a lot of hope for which there is, but also, you know, realism. I don't want, I'm not trying to sell anybody a bridge in Arizona here.
I'm going to proceed on because we have 9 questions and we do have about a half an hour more. So I'm going to zip through these as quickly and efficiently as I can. As we all know, choroideremia, the field of vision grows more narrow as it goes along. And one person is asking, if I'm looking at my television right now, it's more advanced case and I see a narrow, can't see the whole TV anymore, only see a portion of it. Is this kind of scaffolding, this kind of therapy, would that expand it, or how would it work? And if I may ask too, I'm going to combine this with another gal who asks, let me go up. No, excuse, Gabby. No, we already covered that. Go ahead. You answered that one, and I thought I could dovetail it in, but I can't. Go ahead.
Sure. So there's pluses and minuses to every approach. So a scaffold itself has a discrete size to it. In this case, it's a 3 by 5 millimeter, which is about the size of the human macula, or it goes 2 by 4 to 3 by 5, somewhere in there. So yes, your area of treatment is going to be limited. So this is not something that is going to restore your peripheral visual field. This is for folks who have lost nearly all, if not all, of their vision, to restore a portion of their visual field, preferably a portion that's close to their central visual field. It is possible to put in more than one, but that's not going to be the starting point. You're going to put in one and make sure that it doesn't do harm, and make sure that it has some sort of meaningful effect. So you could potentially put in more than one, but because you do have to make an incision in the retina, and there's always a risk that if you make an incision in the retina, and then you place a scaffold like this under there, that what if it doesn't heal? What if the whole retina detaches? You could be worse off than when you started. And that's always a risk when you're doing experimental new therapies. So scaffolds do have that limitation. In gene therapy, it's the limitation of the bleb, or the bubble of fluid with virus that they can put into the retina. And so people are now experimenting with doing more than one injection. So they'll do one injection, creates a bubble, you treat a certain discrete region of the retina, and then let it go. And maybe 2 years later, if it works, you inject someplace else and that sort of thing. So that similar approach could be taken with scaffolds. If we just inject the cells themselves, not completely dissociated, but with some structure in them, we can treat a larger area, but it's still not going to be the whole retina. So that's a very good question, and it speaks to some of the limitations. None of these therapies are going to erase the vestiges of a devastating disease like choroideremia. What we're trying to do is turn back the hands of time in a meaningful fashion, but it's not like going to be you snap your fingers and it's going to be like when you were 5, at least not to start with. That would be unrealistic. And people tell you that, you got to back away and say, this guy's feeding me a line. This guy or gal's feeding me a line.
That's a good point. Managing the expectations is always so important, both from the foundation's perspective and all of our researchers. A question that Ben asks, and it was the one I was trying to incorporate into that last point, is if a person who has really limited vision, low vision left, and this process is successful, is there any kind of work or information that the brain is actually going to be able to see this, to absorb it? Is the information the brain, can it handle it?
Yeah, that should, as I talked about earlier, that once you, and this speaks to me as a pediatric ophthalmologist. So you have a period of visual maturation that lasts, we used to say 8 or 9 years old. Now it's probably into the early double digits of your life. But you learn to see like you learn to walk and you learn to talk. Think about folks who are born deaf. If they don't get hearing aids in time, their speech doesn't develop normally. If they get the hearing aids when they're 7 or 8, they still have speech problems. Okay, because they missed that window of maturation that's important before their brain got hardwired. So we're born with these very plastic brains, and we remain more plastic than we first gave ourselves credit for, even to adulthood, like or semi-adulthood like myself, at least physically adulthood, emotionally maybe not, but but most of that is happening very, very early on in life. And so if you have good, especially central vision, through that very period of visual maturation, you can go blind entirely, but your brain is just waiting for that signal. It's already matured, okay? So, and we know that from classic experiments, and won the Nobel Prize for Hubel and Wiesel. You can do that, and people might not love these types of experiments, and they were done a long, long time ago, But if you put a patch over a kitten's eye for even weeks when it's newborn, and you take the patch off, they'll be blind in that eye. You can put a patch over a fully grown cat's eye for years, and you can take it off and they'll see as good as the day you put the patch on. That's that idea of visual maturation and the critical period of visual maturation early on. So the question you have is a good one, and one that is important for infants that lose vision. So if you lost vision in early infancy and you're not bringing in your therapeutic until you're 18 or above, there's a concern that that is not going to be very helpful. I'll give you another example of how we know about this. And that comes from there's something called a Prakash project that is led by a very prominent scientist who's based at Harvard, but the project is in India, where oftentimes congenital cataracts, which rob children, babies, completely of vision in both eyes. So they're born with these congenital cataracts, and all they can see is white noise because their lenses are pure white inside of their eyes. You then go and remove those cataracts when they're adults, and so now their eyes are perfectly fine. You know, they've got brand new artificial lenses, their retinas are perfectly fine, nothing's wrong with their eyes at all. And you've essentially changed out their windshield, so they should see fine. They still can't see anything. They can see maybe a little bit better, but even though their eyes are perfectly fine, they can't. So that's that idea of severe deprivation of vision early on in life. Since choroideremia patients don't have that, that is not anticipated to be a problem.
Good news. That's very good news. We have several people who are asking about macular holes, and Brent, hello Brent, it's coming to many of ours. He said, with a macular hole in the center of an eye, can that be treated or repaired, followed by applicable treatment of CHM? Or does an eye get the CHM treated, followed by macular hole repair? Or perhaps I just need a good $50,000 pig eye, smiley face.
Yeah, nice. No, I would. So we're kind of chicken and the egg. Some macular holes. Macular holes are actually one of the things we're concerned about. If you take a fragile retina and you peel it off to put in something like a scaffold or new cells, it's possible that center portion pops off, and then you're like, oh my goodness! Again, we go back to the idea that none of these things are guaranteed to be safe. Bad things can happen when you start to do things that have never been done before. So a macular hole, you actually have a piece of the whole retina gone. So that's a more complex scenario than just replacing the cell types themselves. So if you have CHM and a macular hole, and CHM doesn't cause macular holes per se. Macular holes happen to folks that have totally normal retinas. They happen quite a bit for folks that have significant nearsightedness or after cataracts or things like that. But they're kind of like apples and oranges. But it's not a direct result of the choroideremia, but the choroideremia does make the retina thinner. And so little pieces of retina can just kind of pop off sometimes. That is a much more complex scenario. And for cell therapies, we likely would not be, at least initially, looking at patients who have macular holes, because that just makes things harder in terms of the replacement portion of it. For experimental therapies. But there are macular hole surgeries that are available right now that people use, that retina surgeons have done for many, many, many years. A lot of them are successful. And there's some experimental approaches where people are taking pieces of retina that are healthy elsewhere and then transplanting them or moving them within the same eyeball to where that macular hole is. And while it's hard for me to wrap my mind around how that might work, because that requires that whole piece of retinal tissue to hook back up to the brain, as opposed to just the photoreceptors, still, there is some evidence that may be successful. So, yeah, unfortunately, having a macular hole in the context of choroideremia does elevate the complexity of therapeutics.
Mm-hmm. This is a question from Mary. She says, Dr. Gamm, do you see a parallel path for cell and gene therapies, where gene therapy may be recommended for younger, less damaged retinas, and cell therapy for older, more advanced disease states?
Mary, you said it better than I ever did. The answer is yes. just exactly how you said it. Because you don't want to, I mean, you don't want to do gene therapy in somebody who's got nothing left to put the gene into. That would be doing no one a service, right? Nor do you want to take a scaffold or a bunch of cells, lift somebody's retina who's got still very good vision, put those natural cells at risk to put in new houses. Like, you don't want to shove new houses around in houses that are still working, right? Because you could damage the ones that are still working. That's not, my metaphor is falling apart right around there, but hopefully you can follow that. But so there's a time and a place for all these things. That time course is important. Now, if cell therapies over time show safety, then we can walk our way earlier and earlier. And we could feel more confident going into patients who have more remaining vision. Because those new cells might very well have a beneficial effect on the native or host cells. Maybe it will have some sort of a protective effect on them while also replacing the ones that have been lost. It's possible, but again, we have to pick our battles and maximize safety while we're trying to determine just how useful these technologies are going to be for patients.
And can you briefly, again, Dave, talk about where your stem cells come from?
Yeah, so the company and the work that we do they utilize induced pluripotent stem cells. So these are cells that are initially obtained through blood samples, like you might send off for a cholesterol check. So they have in them white blood cells that have nuclei, and then we can use genetic techniques to reprogram them into stem cells. You can also use fibroblasts or small skin punches, about 4 millimeter skin punches. We almost always use blood samples these days. It's just less invasive. Doesn't require a stitch or anything like that, of course. So that's the vast majority of them. A lot of stem cell groups are working with embryonic stem cells, and those are stem cell lines, most of which were developed here at UW-Madison, the H9, H1, or H7 lines. And so those have been the so-called Bush lines, the ones that were approved by George Bush. Those are the ones, at least, that we have here at UW-Madison. But that's not the direction that we're going with for clinical trials.
All right, I'm going to read this verbatim. Ina is asking, I heard that gene therapy not only stops progression, but may revive cells in early stages of deterioration. So a number of retina cells get actually restored during gene therapy. Is that the case? How early a vision loss should be in order for gene therapy to be able to be restored in the cells?
Okay, so I know what you're thinking about are a lot of the RPE65 trials, and those are particularly aimed at the RPE. So recall that the RPE it doesn't detect light. RPE doesn't see light or start vision, but it's a helper cell for photoreceptors. So when you have genetic defects that affect the RPE and RPE alone, now we don't know that's the case for choroideremia. So choroideremia may be an RPE disease, it may be an RPE and a photoreceptor disease. That plays into our other project that I talked about earlier that's being funded through the Randy Wheelock Award. But those that we know are just in the RPE, like RPE65, what happens is they don't go blind because the RPE stops working. They go blind because the photoreceptors that the RPE serves start to stop working or stop working. Now, they're not dead yet. They stop working and they have vision loss, but the cells are still there. So if you do gene therapy and you introduce the new gene and the RPE starts working again, well, then it revs back up and can start to serve the photoreceptors, which aren't quite dead yet. And so you can get some restoration of vision because the photoreceptors haven't died. Once they're dead, they're dead. So gene therapy cannot revive them. I think what people are seeing is that in some rare circumstances, the cells that we thought might be totally dead really still are hanging in there on life support. And that's a good thing. I think, if you'd asked about 5-10 years ago, in a lot of diseases, not just choroideremia, based upon our ability to do live imaging in patients or by looking at histology in eyes that have been donated after death, that we would have said, boy, these cells, you know, are goners. You know, they're well past any hope of getting them to even work a tiny bit. But I think we're found, just like the plasticity in the brain, we're finding that there's probably more there that we can revive than we thought. So that's really, I think, what you're hearing. And that's a good thing. It means that they're not dying as fast as we thought. And the window for gene therapy is probably broader than we initially thought. But again, once those cells are gone, gene therapy is not going to be helpful.
So we have several people asking a very similar question, being, who would be the ideal candidate for a phase 1 clinical trial? And would an individual or a family with several CHMers be better candidates for clinical trials, you know, multiple generations? And then secondly, how do you become a candidate for the trial?
So first of all, right now, there's nothing to become a candidate for right now. So there's nothing to sign up for at this point. Like I said, even though when it's 3 years in advance, there's a lot of things that have to happen between now and then, including FDA approval. And you're not allowed to even talk to patients and say, well, yeah, we'll put you on a list or whatever. That's not even allowed because at this point. So there's nothing that people on this call are missing or that you have to get in line for. And as I said before, it's not a bad thing to be second in line. You don't necessarily want to be first in line. So with regard to multiple people in a family, it really comes down to what are called inclusion and exclusion criteria. So when you develop any new therapy, and that's done in coordination with the FDA, it's a discussion, it's a deliberation in terms of what is it that you hope to do, what is it that you're making, what are the risks that are potentially associated with it. And then that's weighed, so the pluses and minuses, the benefits and risks are weighed. And then it's determined, okay, what level of remaining function do we feel that it's safe to go into? And for a lot of new, never been done types of therapies, it's really the end stage patients. They don't want to risk anyone that could potentially lose a substantial amount of vision that they still have. So you tend to, whether it be a cancer therapy or a Parkinson's therapy or a retinal degeneration therapy, you end up going into the worst of the worst to start with. That's why you have to be aware, you have to know that, because they're also the folks that are perhaps least likely to improve or show great improvement. But you're forced to go into those groups because you have to establish safety. So you want to go into the people who have the least to lose, not the most to gain. So that's important to know, so that when folks come out and they show what might seem very modest positive effects, and everyone's like going, fantastic, that's great. That's really because it allows you then, as long as it's safe, to then go into the cohort of patients or the time frame of patients that you think really will benefit more. So in terms of who can sign up, it's not up to you. You have to ultimately, you ultimately have to agree, but you can't dictate whether you're a candidate or not. It's already dictated. There's a set of criteria. You have to have such and such a visual acuity. You have to have such and such a this. You have to have, from a medical standpoint, you have to be medically stable, to undergo this anesthesia. And, you can't be pregnant. You can't be, during, at the time of the surgery. And all these things kind of go into it. And the people who run the trials, or the people that develop them, like myself, we're not allowed to have any say in that, because we have to be able to be arm's length. And we can have no sense of, well, I know this person and I want to help this person and I don't know you. And so the folks that actually recruit the patients have to be fully removed from the people who are developing it. So that's another aspect to know just about how clinical trials are run. And that's to eliminate bias. Now, that should make a lot of you feel good, because some of you might think, well, if I had enough money and if I had enough connections, I'd be in all these clinical trials. The answer to that is no. For that reason alone, that's the reason why you can't do it, unless it's unscrupulous. There are people that somehow skirt around all of this and are able to do it, and they're strictly motivated by making money, and they can con people with deep pockets into giving them a lot of money to put them in harm's way. And I say, if you're dumb enough to use your money in that regard, then more power to you. because you really have to be buyer beware in all this. And sometimes, you know, a lot of times money doesn't necessarily get you into more trouble than it does help you. So anyway, so folks don't have to worry about, well, I have to jockey for position here or there. What's really important is that you stay involved in organizations like CRF, where when people come forward and say, hey, we want to recruit individuals with retinal degenerative diseases or specifically choroideremia, that they know how to find you. And then you don't have to worry. You don't have to scour the internet. You don't have to worry that, you know, somewhere in Indonesia, there's a cure for blindness. And if only you spent more time on the internet, you'd be able to find it. Believe me, if there was something that was that awesome, it would not be under a rock. Somebody would be trying to make $1 trillion off of it, and they're not going to do that by keeping it hidden. It's going to be shouted at on the mountaintops. So one of the things I like to do in these sorts of discussions is to let folks just kind of relax and realize that, yes, it's important to stay active and involved in organizations like this, to stay aware, you know, not become a hermit somewhere, but at the same time, you don't have to worry that you're missing the boat because, you know, you're not putting enough time or effort into your own personal research for it.
Dr. Gamm, do you think after the procedure is done, is this going to be a permanent restoration, or is it something that would have to be repeated?
We don't know. Our hope is that it would be permanent. Possibilities could be that the body rejects those cells, that the cells themselves don't have the choroideremia genetic defect in them, obviously. So they have the potential to live as long, and developmentally, they're the same as a newborn photoreceptor. So they have the potential to live, to function for 100 plus years. How the body reacts to them, though, we're not quite sure. And there's different ways that we can try to manipulate that. One is to, at least early on, to use steroids and other immunomodulatory agents to try to kind of ease in the new cells, the new wine and the old wineskins, to kind of ease them in so that they're not rejected. However, you don't want to keep people on steroids their whole life. And so as you wean off of that, then you might see some of the cells that are lost. It's also possible that the cells where what we've seen is the cells that do connect tend to stay. The ones that don't tends to go away, but that's fine because I don't want some inert cell hanging around. If it's not gonna work, you know, go home. You know, stay out, get off the job site. So we do have some inkling that might very well be the case. There's also ways that we can kind of get fancy and try to evade the patient's immune system by genetically engineering the cells we put in to be kind of like cloaked so that the body can't see them. But there's a risk to that because let's say there's a problem and the cells aren't functioning right or maybe causing problems. It's kind of a little bit scary to think that you're putting something into a human's body that the human's body is unable to even recognize or know that it's there. It's like giving them the keys to the whole city. So there are a lot of debates going on right now as to whether it's better to have something that the body knows isn't quite you, but we can manage it, versus giving it carte blanche to do whatever it wants. And it will not get rejected, but at the same time, if it decides to be a bad actor, the body has no way of monitoring that.
We have 3 questions left in 10 minutes. Think we can do it?
I can do probably half a question in 10 minutes, but we'll see. I'll try to be quicker.
This is kind of interesting. There's two female carrier questions.
Yeah.
So we're kind of zigging on you a little bit. The first one comes from Dorothy. Would females be candidates for clinical trials?
No.
Why or why not? And if
No.
a female with only light perception is willing to be a candidate for trials of multiple scaffolding because there isn't much sight to lose or much to gain, would you take it? Yes.
So I spoke too soon. This is kind of one of my things. A carrier that has normal vision or reasonably normal vision would never be a candidate, right? Because what do you have to gain? The other thing to know is that if your retinas are fixed, it doesn't fix your genetic predisposition to give that to the next child, okay? So if you put in new cells in your eye, that doesn't have any changes in ovaries or other germ cells. So that's an important point to bring up, because people think, well, if you fix the disease, well, then I can't, transmit it anymore. And the answer to that is no, you can't, because that the code, even though it doesn't affect the germ cells, the sperm and ovaries, it's still contained in them. And we're not doing any surgery there, which of course we don't want to do. Now, there are rare females with choroideremia that do manifest more significant effects of choroideremia. Now, it's always very careful to make sure that you're not talking about something else. So sometimes you can be a carrier for choroideremia, and you have something else that's actually causing your retinal degeneration. So the first thing that, if somebody were to come to me that was a woman with very late stage choroideremia, would be, okay, what's going on here? This is unusual. Being female does not eliminate you from being able to get the therapy, but I have to understand what's going on so that I know that I'm treating the right thing, because that is unusual to have LP vision with choroideremia as a carrier. My first thought is, it's not the choroideremia, it's something else that you have that's causing the LP vision, and you might, and we just have to make sure about that. So.
We do have some people that are in our foundation that have very deteriorated. So I'm really super glad that you spoke on that.
Yeah, and in some cases, there are conditions called Turner syndrome, where women only get one copy of an X chromosome and don't have two, and thus the one that carries the gene mutation, that's all that particular person has. And so it manifests just like it might manifest in a male, because males only have one copy of an X, females have two. So if you're a female that only has one copy, doesn't have a Y chromosome, so you're not a male, but you only have one copy of X, and that's called Turner syndrome, then they will manifest it just like somebody else, just like a male would. So that is one, but like I said before, then you've got two things going on. Right, you have the choroideremia plus Turner syndrome, or an XO. So that's just one example of where that is. Now, there are other, that doesn't mean that if you have severe effects of choroideremia that you necessarily have Turner syndrome. But that's just the complexity of the genetics that you have to wrap your head around before just saying, hey, yeah, just join us.
This next question is super duper good. Manglin asks, would it be possible that the donor iPSCs have other mutations or diseases that may cause other adverse effects in the recipient?
Yep, and they're all screened for it. So we do a screen for all of the lines that we use. We do a screen not only for that, because it's not just other retinal diseases. Now, we do know the donors aren't blind, right? And we know their age, we know their health, we know everything about them, but they're an age. They're like 30 or 40 or whatever they are, they're all different. So maybe they will get something 10 years later that we just don't know about. So we know their history up to that point, and it's a very, very rigorous method. We want to make sure that the cells that we get from them, the blood cells, don't have anything in them. And it's not just blinding disorders. We're interested in tumor genes. We want to make sure that there isn't a predisposition for leukemia, that there isn't a predisposition for growing retinoblastoma or other tumors that might be in the eye. Or, we're also always challenged by the FDA that we can't be 100% sure that the cells we put in might not get outside of the eye. So we have to have a much more rigorous way to screen the cell types that we put in than just blinding disorders. We actually have panels that extend well beyond the eye into other types of disorders that can affect almost every other organ in the body.
One more question. How does X inactivation affect female carriers and their vision?
Yeah, so this is great genetics. So you have two copies. So females that have two copies of X, of the X chromosome, randomly get one of them shut off. It's called, mosaicism, or X-linked inactivation, like you mentioned. So in general, then, what you'll see if you look in a lot of female carriers with choroideremia is kind of this patchy look to them. So oftentimes you can tell by looking at the mom, even though the mom's sitting there going, my vision's normal, although if you do a lot of more sophisticated tests, the mom may think their vision's more normal, but actually they do have some manifestations or some vision loss because of this patchy jigsaw puzzle-like inactivation. So they have pieces of their retina that are more choroideremia-like and pieces that aren't. That's where but when we get back to light perception means the entire retina isn't working, right? Light perception means that the whole retina is knocked out. So that doesn't suggest mosaicism or jigsaw puzzle-y like stuff. And it turns out the brain is really good at filling in the jigsaw puzzle pieces that aren't working as well as the ones that are working well. So it's actually quite remarkable that with this kind of patchwork-like genetic expression with choroideremia that female carriers see as well as they do. But it really kind of comes down to what do you have left? What cells are left? And then what is your function that you have resulting from that? So you may have a lot of cells gone, but if you're still doing really, really well, then we don't want to touch you with cell therapies, right? At least not now, at least not until we're sure that we're not going to do harm. That's going to be better served by gene therapy, hopefully. But if things are far gone and the cells aren't there, then you really have two choices. You replace the cells, or you say to heck with those cells. Let's try to get something else that's more downstream of my retina or my outer retina to work like there were photoreceptors there. That's the prosthetics, that's optogenetics. All right.
You should keep your screen on. I think we both can be, do you still have your screen on? There we go.
No, I just turned it off just so I wouldn't be distracted. So.
We're going to end now, so there won't be any distractions. We have a whole lot of people right now that are saying, thank you, thank you, thank you, thank you.
Oh my goodness, my pleasure, my pleasure. It's such a privilege to work with CRF and folks with the choroideremia in my own clinic, and this just allows me to reach out, and I can sit in my own desk here and have my Diet Coke, and so I appreciate that. Although once things lift up, I'm more than happy to come out and meet people in person. That's always much better.
Well, we are hoping to have you come to our next conference, which we are keeping our fingers crossed will be next June in Rochester. At least that's the hope and the plan.
That's right next door, so that's fine.
Exactly. So again, from the CRF, we just want to say thank you so much. Thanks for being on our bench, being on our team. We're just so appreciative of everything you're doing for us.
Well, that's very nice. And I just want to end it the way I started it, and thank Randy and Randy's family for allowing Randy to do what Randy does, and recruit folks into the choroideremia family. I know that he put a lot of legwork, a lot of time away, a lot of time, tugging on shirt sleeves, and it really did a lot. I know a lot of people who are in this game because of Randy and other folks at CRF. And so, thanks to Randy, thanks to his family. Well,
I think Carol was on today, so I hope she heard that, but I'll let her know.
Yeah.
We want to make sure we say,
so I want to say.
We want to make sure we say thank you again to our sponsors, Allergan and Biogen, and I think that wraps it up. Again, we'll be having this on our website, the recording, and hope you all have a very good weekend, and thanks again.
Great weekend, everybody. Take care. Talk to you soon. Bye.
Thanks, Dr. Gamm.